Welcome to the first in a three-part series of really exciting questions that we're answering. Today's is going to be, I think, very, very interesting, not only for the topic, but because of the speakers that we have with us. The title is CRISPR Opportunities and Challenges Confirmation. Our two speakers are Jodi Halpern and Leah Witkowsky. Jodi is a physician and has her PhD in philosophy. She's a professor here in the School of Public Health at UC Berkeley in Bioethics and Medical Humanities. Her work really brings together really diverse fields of psychiatry, philosophy, affective forecasting, decision neuroscience. She tries to imagine the future and what kinds of influence there will be on this in respect to health possibilities. Her scholarly focus currently is on topics that include emotional decision-making, social dominance and bullying, post-war and social reconciliation, and the ethics of innovative technology. The bullying strikes me as a very appropriate topic for this time in our nation's history. Her more current scholarship focuses on innovative technology change, how we adapt to health losses, how we view our futures, and the trajectory of empathy, curiosity across differences. Jodi has published a book called From Detached Concern to Empathy, Humanizing Medical Practice. This was described by the Journal of the American Medical Association as a seminal work. Her upcoming book is Remaking the Self in the Wake of Illness. And her new book project is Engineering Empathy, which looks at upcoming issues in technology and emotional relationships. Clearly, Jodi has given a lot of thought to this topic about CRISPR. Her colleague, Leah Witkowsky, is a molecular biologist. She received her PhD here at UC Berkeley. So she's had really two careers. Her career as a molecular biologist and her subsequent career in scientific governance, ethics, and multi-perspective engagement. Leah is the executive director of the Kavli Center for Ethics, Science, and the Public at UC Berkeley. She'll describe a little more about that in her talk. Prior to co-creating the Kavli Center, Leah was a policy and engineering manager at IGI, the Innovative Genomics Institute. This is a nonprofit research partnership between UC Berkeley and UC San Francisco. And the project is dedicated to improving and applying genome engineering to solving major world problems. As you can see, both of our speakers are immersed in this topic of the emerging science of CRISPR. Leah and Jodi, I'm going to turn it over to you.
Great. Thanks, John. It's nice to meet you all. As John said, I'm the executive director of the Kavli Center on campus. I don't have a slide about the Kavli Center, but I will just say a few words before I begin. So Jodi and I were part of a team that formed the Kavli Center through sort of a collaborative grant writing process where there was a group of us, 3 scientists who had made major breakthroughs in their sciences, including Jennifer Doudna, and CRISPR was one of them. We also had Stuart Russell, AI, and Jack Gallant, who's done a lot of work in imaging the brain and understanding how the brain translates images and sound and reading those translations out. And the goal for us was really to think about, well, for scientists, especially scientists at a place like Berkeley, where a lot of what we do is basic science or discovery science, how can we start thinking more proactively and importantly, training the next generation of scientists to be better thinkers about the ethics of their work and how to involve the public and important stakeholders and communities in the process of thinking about what work should be done and how that work should be applied. So we founded this center, which I am now the executive director of, and Jodi is one of our core faculty and on the steering committee. So Jodi and I have spent, I guess now, maybe 7 years working together, and now more formally through this role. So you'll notice that we may build on each other's thoughts as we go through this. So I'm gonna get started. We decided to split this today. I'm gonna give you some background on CRISPR. I know some of you may have heard of CRISPR, some of you may not have, or perhaps you heard of it, but you're not quite sure how it works. So I thought I would start from the basics, how it works, how it was discovered, and then touch on some applications, so some of the potential opportunities that arise directly out of the mechanism of CRISPR. And then I'm going to highlight very broadly some of the types of ethical questions that you might expect to come up when you're using a technology like this. And then I'm going to turn it over to Jodi to dig into one example of an ethical issue, which is germline gene editing, and I'll explain what that is a little later. So to get started, let me walk you through how it works. And to do that, I'm actually going to tell you the story of CRISPR. So this goes back over 35 years ago. So even though CRISPR seems like this hot new technology, it is, but this whole thing started 35 years ago when scientists who were studying bacteria and had just kind of gotten their hands on the ability to do sequencing of DNA were studying this bacteria, E. coli, and we're looking at its DNA. And I think most of us here may be familiar that all living things have DNA. The DNA encodes the function of proteins and the ways that the cells function and what our bodies are composed of and how they work. So bacteria as a living thing also has DNA, and it also has a genome. So 35 years ago in 87, more than 35 years ago, some scientists in Japan were studying sequencing bits of this bacterial genome, and they were looking at this one type of gene. And right next to the gene, they saw what I'm depicting here. So here's sort of the caricature of DNA, and then here's sort of a color block way of depicting it. So they looked at the genome, and next to the gene that they were studying was this long repeated sequence. And these were short little bits of DNA. So now DNA, usually if it's encoding a function for your body, in other words, if it's a gene that's encoding a protein, it has a really characteristic feature. It has a start and it has an end, and those things are part of the DNA. And so we know it's a gene because it has a start and an end. When they were looking at this, they saw this weird repeated sequence. It was quite long. All of the repeats were really short, and they had these random little inserts in between them. So the repeats here are shown in white, and then spacers of random sequence, each one different between these repeats. They knew this wasn't a gene, but they didn't know what it was. And it took over 20 years after discovering this region to figure out what this actually does. And part of the reason it took that long is because we improved our methods for sequencing, and we started to amass a lot of data of sequences of various kinds of organisms out in the world. And so in 2005, a researcher and a team started looking at, well, what might these random sequences match to? Are they similar to anything else that we have in our databases? And what they found is that, in fact, those sequences matched not other bacteria, but viruses. And, oh, I skipped this part, my bad. So just to give you a sense of why do we call it CRISPR? So the acronym is clustered, regularly interspaced, short palindromic repeats. And this is essentially just a description of this weird bit of DNA that was discovered. So it's a cluster of regularly interspaced. So there's these repeats that are regularly interspaced with these spacers. Short, they're little, palindromic, these repeats happen to be the same front and back, repeats. That's CRISPR, okay? So then in 2005, yes, they matched these seemingly random spacers to sequences from viruses that infect bacteria. So just like humans, viruses can get an infection, and the way that it works, and this is actually a electron micrograph image. So these are real bacterial cells, and real bacteriophages is what you call a bacterial virus infecting. So they infect, they inject their DNA, they hijack the cell's machinery to make more viruses, and then the viruses explode out of the cell, killing the cell. Right? So it turns out that those seemingly random spacers matched DNA from the virus. So here I'm showing you essentially from that exact image, a real electron micrograph image. Here's more of a drawing of it, of a virus infecting a bacterial cell. It injects its DNA into the bacterial cell wall. And in fact, here I'm showing you with the pink that for some of these viruses, it matches one spacer, not every spacer, but one virus will match one spacer. A different virus might match a different spacer. So some of you may already know the answer to this, but what do you think this might be doing? Why would a cell have a region where it has little bits of DNA that match different viruses? Does anyone have any thoughts or ideas? If anyone knows, you're welcome to share that as well. Built-in redundancy for repair. Interesting. Repair what? To repair the virus? Okay, so one person says virus has previously attacked the cell. Yes. Strips of DNA. Yep, it's little copies that match the viral DNA in the actual bacterial DNA. So one of you has guessed, and I don't know if you've heard this already or not, but scientists saw this. They saw when they sequenced this region that it matched a series of viruses and started to hypothesize that, well, what if the reason that it matches the virus is that it is encoding the viral sequences as a memory of past infections? And so, in fact, that is what it's doing. And just in the next 2 years after first discovering what these matched, people did experiments to show that, in fact, yes, the reason that it matches is that when a virus infects the bacteria, a little chunk of this virus DNA is actually copied and inserted into this region. And by doing that, when the cell divides, it gives its genome to its progeny. And so now all progeny of this bacteria that had seen this infection at one point will have a copy to be able to identify that virus again in the future. And the way that it does that is it reads out its own genome in the CRISPR array, the CRISPR area. It copies this out and turns it into this thing called a guide RNA. So RNA is just a smaller, more transportable version of DNA, but it's essentially the same thing. So it uses this as sort of the blueprint. It makes a copy of the guide RNA. It loads it into this protein complex that is constantly surveying the bacterial cell. And at some point in the future, when the descendants of this original bacteria encounter another virus of that same kind, this constantly surveying complex will match exactly to the sequence of the invading virus that its ancestor had seen, and it will create a cut in that DNA. And by cutting the DNA, it's essentially destroying any chance that that bacteria has of hijacking the cell and replicating. So this is a molecular memory of past infections, and it is a bacterial immune system. So this is CRISPR. Okay, so why? Like what? So what, right? The exciting thing, I would argue, and I think what many people have really heard about, is that not only is this just cool from a science perspective, turns out bacteria have immune systems like people. That's unusual, right? That's cool. It's cool, and it can be repurposed. So that is the major discovery that Jennifer Doudna, who is a faculty here at Berkeley and a Nobel Prize winner, as well as her collaborator, Emmanuelle Charpentier, the thing that they did that was so monumental was not that they discovered CRISPR. That happened 35 years ago. What they did is they said, well, what if, first of all, they described how this works in detail. And then they said, you know, all that this is a protein that has a tiny bit of RNA that matches exactly to a target DNA, and it cuts it. What if instead of matching viral DNA, what if we told this piece of RNA to match something completely different, like a gene that we care about that we want to cut? So they did that experiment, and they showed that, in fact, you can, simply by changing the sequence of the guide RNA, you can redirect this complex to cut any sequence you want. And so now that went from just being a cool biology discovery to a molecular tool that you could use to engineer DNA in cells or outside of cells. So this is kind of the major breakthrough that has led to some of the fastest moving biology science that's applied, I think, that we've seen in many decades. And the reason it's so fast moving and so fast paced is because it is incredibly easy to use. So just to give you a sense of how you would do this. So let's imagine you are a CRISPR scientist, or maybe you're a clinical researcher. Maybe you want to try and cure a disease that you know is a heritable disease that's caused by a single point mutation in a gene. So here I'm showing you an actual sequence from a genome. And what this is the sequence encoding the hemoglobin molecules that you need to carry oxygen around in your blood. And this red letter, so these are the DNA, the two strands of the DNA, and these are the letters that show you the sequence. These two letters are a mutation that occurs in people that are living with sickle cell disease. Their sickle cell is caused by a single incorrect letter in this one gene. So if you were a scientist and you wanted to fix that, let's say, or target that, what you would do is you would first find an area where your guide can land, and that's called a PAM, and we can talk about that if you want. And then you would just simply count 20 base pairs up from that and copy, literally take your spacer in the sequence that's on the website of the human genome that's uploaded to NIH. You copy that sequence, you put it into another window in a computer program, and you say, I want to target this. You send that off to be created in a synthesis lab. So they will synthesize a guide RNA, so that little piece of RNA that got loaded. You will give it this sequence. It will make that exact sequence, spit it out, and now you have your guide. And that guide will guide the cutting complex to whatever sequence you programmed it to. You load it into that cutting complex, and it searches the genome, and when it finds an exact match, it opens the DNA, and that opening exact match causes the cut. So this is how genome editing works, and here, this is exactly a cut that is being used in clinical trials right now to make changes to a sickle cell disease gene. So let's see where I am. So as I kind of just alluded to, part of the reason that this is so exciting is that being able to target a specific gene, a specific part of a genome, and make a cut there means that we can disrupt a gene and we can change it. And so even things that are a single letter, like sickle cell, can make a really big difference for somebody. So the last piece of information to give you about how this works is what happens after the cut. So traditionally, right, just taking the CRISPR system out of the bacterial immune system and repurposing it to be a genome editing tool, what you essentially do is you decide, I have a gene that I want to target. I know the sequence of it. I will make my guide. I will load it into my cutting complex, and it will cut the gene. So what happens after the cut is the second step. And that second step of editing is actually taking advantage of the natural repair processes that your cells already have. So DNA is incredibly valuable and important. You really don't want your DNA to be destroyed. And so throughout evolution, virtually all living cells have the ability to repair DNA because it's so critical to the survival of the species. And so what we do as genome editors is we take advantage of those natural repair pathways. We make the cut, and then we either target that cut for this pathway or this pathway. So let me tell you what those are. The non-homologous end joining pathway is essentially the pathway in your cells that say, oh no, the DNA is broken, we're going to stitch it back together. And it literally just puts it back together, the two ends. Most of the time, this works well. And actually, this is something that happens constantly in your body. We're exposed to mutagens all the time. The sun, for example, is constantly bombarding your DNA and creating breaks. Your cell is constantly using this pathway, just putting those ends back together. And it almost always works, okay? But if you have something in your cell that keeps cutting it over and over and over every time it repairs correctly, every once in a while, it'll repair incorrectly. And now that that repair missed a DNA or it overlapped wrong, it's no longer a match to your cutting complex, to your guide. And what you've done is you've created a new mutation that didn't exist. So that mutation often disrupts a gene. Okay, so you cut it, it repairs wrong, and now you've messed up the whole sequence of the gene and it no longer is going to be used or read. So this is what we do if we try to turn off a gene. The other repair pathway that researchers will use is called homology-directed repair. And the way that that works is, of course, we all have more than one copy of a gene in our cell. We have genes from our mom and genes from our dad. And the great thing about that is if one of them gets messed up, you can kind of infer what it probably said by the exact copy that came from your other parent, right? So homology just means that it's matching the sequence. So it naturally would use the other copy of the gene as a template to repair the broken one. So this is the other pathway that we can use. And the nice thing about that is that it doesn't just disrupt a gene by making a mistake. Turns out you can, instead of using your natural copy from your other parent, you can give a little piece of DNA that has the sequence that you want to be the new sequence. And that template gets used instead. So when it repairs, it will write in to the cut all of this new information because you gave it this kind of mock copy of the gene. So using this method, we can insert new sequences. We can change a single base pair because template we're giving it just has one letter change. We could change multiple letters. This is a lot more control in the way that the gene gets changed. So this is the overview. And now that you kind of have a sense of how it works, what doors does that open up for the opportunities? So if it has DNA, which all living things do, we can edit it. And it's hard to understate how incredible that is. Most things that we do in a lab until CRISPR work in the lab. And then you try and take them out in the real world and they don't work that well. Or maybe they work on your model organism. But once you move out of the model organism, it doesn't work that well. For whatever reason, CRISPR has just worked. Sometimes it takes some troubleshooting. You have to grow your methods and your knowledge about how to do it. But every organism that it's been tried in, it has worked. So this is what I'm showing here are what you would call model organisms. And this is organisms that for decades before CRISPR, this is how we understood genes. This is how we understood biology, because these are the organisms where we could actually manipulate the genes. But now with CRISPR, we can manipulate genes in any living thing. So we no longer have, 1, 2, 3, 4, 5, 6, 7 model organisms. Now everything can be edited. Everything is a model organism. So this means we can use it to study things, to just learn more about how things work. This is a basic science tool. But it also means that we can do things like, edit crops for food production to protect against climate change. We could, if we know, for example, that there's a species that is threatened for climate change. So these are things that I'm showing here are actual projects that scientists are actively working on. And the one on this side is in California. There's a type of pine that is being threatened by disease, and that disease is caused by changes in the climate. And it turns out that that type of pine exists in many regions across North America. There's subspecies in those different regions that have different genes. And so if we knew a species that has normally evolved in a hot climate, if we can figure out what kinds of genes give that subspecies the protective advantage in a hot climate, we could port that into the Northwest pines or the California pines so that we preserve the genetic diversity, because it's still the California pine, but now it has this one extra gene to protect against climate change. And that's the same concept with the coral reefs. They're studying that as well. And again, the whole reason to change the DNA rather than just bring in the new species is that you don't want monoculture. You want to maintain the original diversity of species, but just give it one little bit of advantage. Crops are another obvious thing. There's a lot of work going on to protect against various droughts, actually flood as well. There's also disease and pest management. And then there's the whole host of human diseases that could be potentially targeted using genome editing, most of which we would be a little bit more obvious in the sense, if you have a disease that is heritable, so it is caused by a gene or mutation in a gene, then in theory, it could be targeted and fixed with genome editing. I say in theory because it takes a lot of work to develop that and to test it in clinical trials and make sure it's safe. But here are some examples of real work that's happening. This is sickle cell disease. The first CRISPR treatment for sickle cell was approved by the FDA, I think this year or last year. This is a project working on degenerative eye disorders. And then there's a whole host of research going on using CRISPR to improve our immune system to better fight cancer. So these are the opportunities, I would say, that CRISPR presents. So it's all kinds of new questions we can ask in basic science, but there's also all kinds of new applications that have real world benefits that we can do. So what's potentially concerning about that? It turns out there's a lot. So this next part, I'm just going to give you a quick overview, and then I'm going to turn it to Jodi to dig in a little deeper on one specific issue. So before I do that, maybe I should pause and say, are there any questions about this first part, about the science and about the opportunities?
Lee, Annette asked, does one cut work for both strands, or are two cuts needed?
That's a really great question. So it turns out that the natural enzyme that we use will cut both strands. Some people have engineered it to only be able to cut one strand, and that's actually a strategy for if you're trying to make a cut on one strand and use that to target for repair. It turns out there's a different repair pathway for a single strand cut. There's ways that you can do that. So you could cut one strand on one side of the DNA, and then hundreds of base pairs over, you could cut the other strand and try and kind of pop out the whole section. There's ways that people have used engineering of that enzyme, that complex that does the cutting, to redirect it and change it a little bit from its natural function so that we can have a whole variety of types of editing happening. I hope that answered your question.
Malia asked, could you use CRISPR to edit, say, the ApoE4 gene that is?
Yes, you could, and people are working on that. And actually, when I had first put this deck together, I had put a section of potential case studies that we could discuss as a group, and one of them was ApoE4, because there are, I think, ethical questions around that. And I think I may touch on it a little bit here in a second, but if we have time, we can discuss that at the end.
For those not familiar with ApoE4 association with Alzheimer's disease, Chuck asked, time frame after making a cut that dispersion occurs.
So actually, this was one of the things I studied in my PhD, was how long it takes to do this in the cell. It's very fast, on the order of hours, which is, for many things in biology, fast. It may not sound that fast to you, but it is. Yeah. I hope that.
That's all the questions we have right
Okay.
now.
All right, so let's think a little bit about, given the capabilities, what sorts of risks or dilemmas might we expect? So I generally like to categorize these things in these three ways. I don't know how academically rigorous this is, but this is how my brain thinks about it. So the risks and dilemmas that stem from these different methods of editing can be thought of as technical risks, philosophical or moral risks, and then societal risks. So what do I mean by that? So technical risks are things having to do with the science itself and the capabilities of the scientists and the methods. So some things that we already know are questions still that we're still trying to figure out. One of them is off-target effects. And what this means is, here I'm showing you another way of showing DNA. So here's this long bit of DNA. Here's the CRISPR complex with the guide RNA and the cutting complex. And you've intentionally targeted to cut this gene. So what are the chances that maybe some of the time it cuts something else? So that's a question. And it's actually very accurate in its cutting. It's what you call, it has high fidelity. It's not very promiscuous. But it does every once in a while bond to an incorrect sequence. And that is depicted here. So essentially, this is maybe the perfect match of the sequence. And then there's all of these other in your genome imperfect matches. And how, what percent of the time are you binding and cutting to those imperfect matches? So that's one example of a technical risk, because we definitely don't want to go around cutting up the whole genome by accident. Another example would be immune response. So this is a bacterial protein system, and you're putting it potentially in people. Is that going to cause an immune response that could be dangerous? Maybe. The other question is persistence. So how long, if I am trying to make a therapeutic and I'm injecting this complex, is it just going to live in the cells forever? Is it going to get, transported to other cells? Do I want it to be in my cells forever? And how well does it work? So these are all technical risks. And these are all overcomable. And I think that matters, because a lot of the discussion from scientists around ethical dilemmas often is focused around the technical risks. What would happen if we had all these off-target cuttings? These are important and critical things to be thinking about when you're making a therapy, but they are things that we will eventually overcome. There are methods that we can use. What I'm showing you here, this is sort of the classic CRISPR complex. This is directly out of the bacteria. But like I mentioned in one of the questions, people have gone and engineered the CRISPR system itself to have all these other functions now. So now there's all these other ways that you can actually make it even more discriminating on off targets. So people have engineered it so it almost never has off targets. People have added additional things to it. So it has, maybe it's not cutting anymore at all, but instead it's recruiting other proteins to do other things. So over time, all of these technical risks are addressable. So what does that leave? I think what it leaves are these really difficult questions that don't have scientific answers. So if we can, should we? So now we have, this door has been theoretically open to all kinds of things that we couldn't do before. Should we do all of those things? What are unintended consequences? I think especially what should our relationship with nature look like? I'm talking a lot about the therapeutic applications, but as I showed you, there's all kinds of different environmental and nature-related applications that have nothing to do with humans other than we benefit from them. Should we be engineering all of the organisms around us? Who does an application benefit? Who does it harm? What is responsible use and governance? So these are actually the questions that I think Jodi and I are more interested in these days and are related to why we founded the Kavli Center. This is the kind of stuff that we want to encourage scientists to think more about and to start learning techniques for how to think about them. Oh, it's not working. And I don't want to take up all the time, so I'm just going to skip through a couple things. So for this other category, the non-technical stuff, you could think of things as philosophical and moral, which might have to do sometimes more with what do we owe other people, individuals, how can we think about risk benefit? And then there's also societal level questions of how should we behave as a society, not just as individuals to each other. So some examples are, if we are using this for therapies, is it going to benefit everyone? So is there a risk that we could exacerbate health inequities? This is a real concern. And the reason being that these are going to be the most expensive drugs that have ever been approved, ever. So will they even be covered for enough people by our insurance? And if they are, is that going to take all the money away from the insurance companies for other kind of more mundane healthcare coverage that we really need as a society? There's questions about what are we treating in the first place? So, things like sickle cell disease and really debilitating diseases might seem obvious candidates, like we should treat those. But then at what point, do we know that something is definitely going to be a high risk, serious disease just based on the sequence? Sometimes there's a range of possibilities. You may or may not develop a disease. So if you have the BRCA gene for breast cancer, it's a pretty high % chance, but it's not guaranteed. So there's many more genomic bases of disease like that have a different range of risks and % likelihoods. And then there's the question of enhancement and prevention of disease versus treating disease. So these are all kind of ethical and societal questions. Again, with the disease question, what is a disease in the first place? What should be cured and what becomes just part of our spectrum of diversity of lived experiences? So people who are deaf, some parts of that deaf community have created a whole other culture around deafness. So there's a language associated with deafness. If we eradicate deafness that is caused by a genetic predisposition to deafness, then we are also eradicating that culture. Is that something we should do? Somebody asked about Alzheimer's. So is Alzheimer's a disease that we should be eradicating? Maybe, probably, but this is something in the brain. So at what point does changing the genetics of your brain bring into questions about what it is to be human, how we think about our brains being ourselves versus brains being pathological? What are the risks of doing that? And then, of course, Alzheimer's is something that happens later in life. So at what point do we know for sure somebody is going to get Alzheimer's and then we want to prevent that versus waiting until they get it and treating them? And then the last bit in the human section, I'll talk about again, but these are questions about creating engineered offspring, essentially. And just to make sure I'm touching on everything, so those are the human issues and just some of them. And again, there's all kinds of other applications that are non-human applications that also come with ethical challenges. And some of those are very similar in nature. So one example would be engineering microbes in the environment. So let's say we knew, and it turns out we do know, that microbes play a really big role in sequestering or not carbon from the atmosphere. So we could, in theory, figure out which genes and which microbes are involved in that process and engineer them to be more efficient at sequestering carbon. That's something that's out in the world. Microbes don't obey borders. They will multiply, they will spread. So if my community decides to do it, I'm kind of inherently making a decision for everybody else because those microbes will spread. How do we manage that? Whose decision is it? Who stands to benefit from that? Again, there's also differing views on values of different species. So this is all kind of just a general, trying to get you to realize that there's many opportunities, but there's also with them many questions that are not easy to answer. So we can talk about some of those if we have time, but the one that we really want to dig in on for the rest of this session is germline genome editing. And this is, if you've heard of any ethical things with CRISPR, this is probably what you've heard of. So what is germline genome editing? It turns out, right, that your cells in your body all share the same DNA, all the same genes. At some point during the development from an egg and a sperm becoming an embryo, those cells that all have the same genes start to differentiate, is what we call them. So at some point, those cells start to say, I am now going to become a skin cell, or I am going to become an eye cell. And those are what you would call somatic cells at that point. So they differentiate, they know they're going to be a skin cell, an eye cell, and they cannot create a new human just from themselves, even though they have the same genes. Another portion of those cells maintain that totipotency, where they're able to become any cell in the future. Those are your germ cells. Those are the cells that become your sperm or your egg. Those are also things like your gonads, early stage embryos. So these are your germ cells, meaning that those cells carry that information just like all your other cells, but they also pass that information on to all other progeny downstream. So this matters because when we think about the therapies that are currently being pursued with CRISPR, like sickle cell disease, these are somatic cell therapies. These are therapies that are treating differentiated cells or partially differentiated. Stem cells still are not germ cells. So you're treating your blood system, right? If you're doing sickle cell, you're not treating, whoop, I just lost it. You're not treating your entire body. And let me see what happened here. One moment. There we go. Sorry about that. There's interest and questions on whether we should move beyond somatic cells. So you treat a somatic cell in a person. When that person dies, that change dies with them. If you treat a germline cell with genome editing, that will get passed on if they reproduce to all future progeny. And just how might you do that? This is something that was actually done in much shock and horror to most of the scientific community in 2018. And what they did was they took an egg and a sperm and did in vitro fertilization. And when they're doing that part, which we've done for decades now, in vitro fertilization is a standard technique at this point. During that process, they also included CRISPR in the injection of the sperm. So it's just one additional step. And when the sperm meets the egg, the CRISPR cuts the DNA from the sperm and the DNA from the egg. That's the idea. And so those cuts in that one cell embryo divide and copy to become all the cells in the baby, including the baby's germ cells. So the babies will pass on that edit as well. So at this point, why might we be concerned about this and what should we do about it? There's a paper that Jodi led in which I collaborated along with some of my colleagues, and I'm going to turn it to you, Jodi, at this point.
All right. So I'm so lucky that we have this fantastic background that Lee just provided. what we're going to do now is I'm just going to go over what Lee and I and our colleagues, the work we did in this paper and why we did it, because we were in the middle of a very important societal moment that Lee mentioned that I'll get into. So I'm only talking about germline genome editing, which Lee just explained. And it's a very important thing because, as Lee said, it affects the future progeny of everyone who's edited. So it really affects humans and overall. I don't have to explain almost anything on this slide except the last bullet, public health, societal ethics. It's what I just said. Since germline editing affects everyone, it's different than just an individual medical decision. It has to be thought of from a societal point of view, as well as from the individual's point of view. Okay, so down at the bottom of the slide on the left, well, I'll start up at the upper left. As of 2016, there was a world consensus put into language in the 2016 International Summit that it would be irresponsible to proceed with any clinical use of germline editing at that point. And everyone, all the major scientists, all the leading society and ethics and science thinkers, all governance structures agreed that we were not ready for germline gene editing. And actually, I love Lee's division. We weren't ready technically, we weren't ready philosophically, and we weren't ready in terms of societal interests. And there was absolute agreement, at least publicly, about that. And yet a scientist went ahead and did it. And this scientist is a physicist. You'll hear that part of my issue with all of this is that the people who actually have the tools for this are often trained in physics or chemistry, and they have no training, as they might have if they were physician scientists, in the rights of human subjects, ethical obligations, and human research. So this person, in China, his name is, he's a PhD physicist who actually trained also at Rice University and Stanford here. His name is He Jiankui, but he goes by JK, and I'm just going to say that to save time. So JK announced in Nature, and it was covered in Nature, the journal Nature, that he had edited genome-edited twins. And there was really quickly a big public outcry over that internationally because it broke this norm that we were not ready technically, ethically, or as societies. What I want to do is talk about 3 sets of ethical challenges that at the time, there's even more I think we could think of since then, but that his exact approach raised. One of them, which does not get a lot of focus, but it's a big interest of mine and Lee's, is just the way he violated professional research ethics, because we are training at Kavli this new generation of scientists, and we want them to think about what they do as professionals. The second is the ambiguity of the edit he did between treatment and enhancement. Lee raised that, but I'll go back to that. And then the third is that there are no, and at that point, were absolutely no clear and robust international regulatory frameworks. So those are the 3 things I'll get into now. So first, the failure of professional research ethics. First of all, as I said, JK was a biophysicist with no prior research with human subjects and no training in that area. And what happened in terms of the actual research ethics is he put out an ad for couples in China where the father was HIV positive and the person who wanted to be the mother was not. And they wanted to have a child biologically, and they were concerned about transmitting HIV to their baby. And he enrolled them for this trial. They were usually people who could not afford a procedure that he used before he did the germline gene editing, which is sperm washing. When you get sperm washing of the dad who's HIV positive, that's a pretty good protection that the embryo and child that develops will not have HIV. So if they simply had sperm washing, which is very safe, they could have been protected from their child being born with HIV. But the problem is that they couldn't afford it. They couldn't afford the sperm washing. So this couple signed up because this was, as far as we know, one of the possible criteria was this is the only way they could even get sperm washing. What we really don't know is if they even understood that that was sufficient to protect their baby, because along with that, giving them the sperm washing, JK did the first ever in human germline gene edit. And that was a high risk procedure technically, because, for example, we didn't know about the immunologic or other features, but we also knew, and it turned out that it was likely that not all the cells would get edited, and there would be a mosaic pattern, which can cause its own issues in the embryo and other things. So we have no idea if they understood any of those risks, but we do have the impression, at least from the way J.K. talks about it, he presents it as this is what they needed to do to not transmit HIV to the embryo, but it's not. Sperm washing would have been sufficient. So that's a big ethical no-no, is to sign people up for a very high-risk procedure when they don't really understand why they need it or what the risks involved are. And so you can say that this violates really the major criteria for research ethics, which is respect for persons, where people have to have understanding and voluntariness, because it was their only way to get something of great importance to them. It violates beneficence because of the safety risks, and it violates justice because it was poorer people who couldn't afford sperm washing in China who he was registering for this trial. Okay, the second issue I raised ethically, I'll just go back to show you. These are the, sorry, I'm going to talk about professional research ethics. We just did that. Now I'm talking about the ambiguity between treatment and enhancement. Basically, what the edit that J.K. did on this embryo, on these embryos, was of the CCR5 gene. The CCR5 gene confers a great deal of almost perfect immunity from certain kinds of HIV if you're exposed to it. So if you have an edit of your CCR5 gene and you grow up to be a sexually active person, when you have sexual contact with someone who's HIV positive, if you have that the correct gene for that corrected gene, you'll actually be protected from the transmission of HIV. So it's an interesting question whether that's treatment or enhancement, because these, as I said, they would be born with no disease, and they were born with no disease, the twins. The sperm washing took care of that, but the gene then made them at lower risk of getting HIV when they became sexually active people. And so I think there's a good argument you can look at that as enhancement, but it's worth arguing about. But because we do have at least some knowledge of how to protect ourselves from HIV, and there's widespread condom availability in China. It's not out of cost reach for people, and we also have treatments for HIV. But was it a very serious disease that we were using the germline edit for? I think an interesting example of the ambiguity between treatment and enhancement, and to see how, when I say it's context dependent, it basically changes over time as we get used to more and more ways of promoting our health. So for example, many years ago, anti-cholesterol meds were thought of as enhancement because most people had, didn't think and have access to a way to actually prevent themselves from developing atherosclerotic heart disease. But then, we've all gotten used to the thought, which I think is very good, that we can have anti-cholesterol medications widely used to prevent very serious heart disease. So even though it's prevention, it's not treatment, but it's not enhancement. So prevention in this sense of a very serious disease seems to have the same moral status as treatment once it becomes generally used. And what's interesting is we actually have a gene that we can edit, the PCSK9 gene. And if we edit that, that will render people immune to the effects of cholesterol, just as the CCR5 gene rendered people immune to HIV. So if we did germline gene editing for PCSK9, we could quickly create a population that would not be at risk of atherosclerotic heart disease, which I think probably all of us think would be very beneficial, and a serious removal of a very serious disease burden. So it's very interesting because we don't have that now. So if someone's in an experimental trial using that for somatic cell edits, not germline edits, we don't have it for that, but there are trials where research studies where it can be used for your somatic cells. We don't necessarily think of that as enhancement, even though most people don't have access to it. And then we also don't really have yet really solid international regulatory standards. That was my third ethical issue. But we did write about in this article that Lee showed you that we did after JK's edits. At that time, his edits were at the time that his edits were done, two influential reports were in the making. which had specifically addressed germline gene editing. One of them was the NASEM, is the National Academy of Science, Engineering, and Medicine in the United States. And they had put out a consensus report on genome editing. Victor Tsao is the president of NASEM. I actually got to be on the panel with him, the international media panel after JK did the edits. It was Victor Tsao and me. If you want to watch it, it's on YouTube. I'll put a little plug in where we address the ethics of what JK did. But this, I'm going to talk about what this report said. And then in 2018, the UK Nuffield Council put out a report. And what I'm really using the reports to do is what we did in the articles. We showed how they had different approaches to how to think about the ethics. And I'll go over those approaches and see why they're both necessary but not sufficient. So first, the National Academy of Science, that's this panel, NASEM Genome Editing Consensus Report, the National Academy of Science criteria. They said germline gene editing could be appropriate when there was the absence of reasonable alternatives, when it was restricted to preventing a serious disease or condition, and very importantly, with continued reassessment of both health and societal benefits and risks, with broad ongoing participation and input by the public. So they called for polling the public or some form of public engagement. And they also called for reliable oversight mechanisms. So that's the focus of the National Academy of Sciences in the US. The Nuffield report from the UK, 2018 report, had a somewhat different approach. It emphasized certain principles. It said that germline gene editing would have to uphold principles of social justice and solidarity. So again, they're seeing it very much as a technology that is a societal technology and not just for an individual's health decisions. So it shouldn't be used, even if an individual could very much benefit, unless it could be expected to not to increase disadvantage, discrimination, or division in society. And that's very important because Lee showed you the slide that the cost of these treatments is astronomical. So the odds that it would be distributed equally are very, very low. So right away, it's kind of clashing with that Nuffield principle. And the second principle that they stated is that heritable genome editing interventions, germline genome editing, should be permitted only when the impact on those who are vulnerable to adverse effects, including stigmatization, British spelling, and discrimination, might thereby be increased, has been assessed and mitigated. And basically, and not without consultation with those groups. So basically, this is saying, like Lee mentioned, the deaf community, that if it becomes the norm to do germline gene editing for certain disabilities, for example, then it could become more stigmatized to have, for example, deafness. Not only might deaf culture be imperiled, but if people did not have the money and funds to have gene editing in their family lines, their children might be discriminated against because we wouldn't necessarily have the societal agreement to fund services for the deaf. Uh-oh. Okay. So the two different approaches of these guidelines, they actually overlap quite a bit, but two major different emphases is the US one really emphasized public engagement, and the UK one, while it also did actually call for public engagement, gave very specific principles of social solidarity, social justice. So let's look over those two approaches. Can we come up with a good international governance by really looking to public engagement? Well, first of all, I want to say, as Lee mentioned, with the Kavli Center, we both think public engagement is very important. So in no way am I saying we don't need public engagement, but the question is it going to get us really there, all the way there, to internationally agreed upon ethical guidelines? Well, we think public engagement is necessary for many ethical reasons. First of all, it captures dissenting views. People really do have different views on this. Secondly, it establishes conduits of transparency and expanded understanding. This is really important. It's a big part of why Lee and I have worked to have scientists think about ethics early on in their development of innovation, but scientists alone by no means should make the decisions for society. And society doesn't really always know what's going on in the innovative science, or even if it's translation into products, until it's basically too late for society to have an impact on those products not being put out to the public. So we really need the public to see transparently what is going on in highly innovative science and to really understand what's at stake. And then there's a third distinct but important ethical reason to have public engagement, which is to give people voice, democracy, that we need procedural justice and we need fairness and inclusion of the voices of people from across society. And fourth, but not in order of importance, all of this public engagement not only respects society and justice, but it respects the autonomy of individual citizens. So all of this says it's important, but why do we think it's insufficient? Well, in practice, first of all, we have yet to really have a science of public engagement that really guides us into which forms of public engagement are more reliable and more sustainable in their governance structures than others, that society will be sort of happiest with and feel did the most productive work over the long run. But second of all, we even have obvious framing problems in a lot of the societal public engagement that goes on. So for example, the National Academy of Science body that I mentioned and their report, they had at the end of it that they did a poll of the public and they asked the public whether they would approve germline gene editing for serious genetic disorder, and said, more than 50% of the public said yes. But what they did in that question, the way they framed the question, they said nothing about the cost. So the implication is that, and what we believe is that the people who said yes, they might want it for a very serious disease, were envisioning it if their family member had it. But they weren't thinking about that they may never be able to afford it, and that may be something that would take a long, long time to be affordable to anybody but very wealthy people. So that's not really a fair way to ask the question. And second of all, there's profound questions about who are we really engaging in the public to make the ultimate decisions about governance. For example, sometimes we poll people with disease, and we actually just had a great discussion with our center yesterday about the citizens jury on this very question of germline gene editing in the UK, funded by the Wellcome Trust. And they did a very, I think, rigorous process of public engagement, but everyone that was invited to participate had to have a genetic disorder themselves or have an immediate family member with a genetic disorder. So anything that those folks produce will be very important because, first of all, epistemically, they know what it's like to have various genetic disorders, although they all have different ones, which is interesting too. But secondly, from a justice point of view, we do want to include the voices of people affected by the disease, like we'd want to include the deaf community in deciding about whether we should have germline gene editing for deafness. But the interesting thing is that the only voices that really should dominate, because as Lee also mentioned earlier, with expensive treatments, we're using health dollars that maybe could go for preventing malnutrition. In this country, we have a majority of children in certain parts of the country that have poor nutrition, and people don't even get screened for high blood pressure, or we have very basic, easily preventable health problems, diabetes in this country, that just health dollars that were used for public health interventions could go a long way. Is that more important than treating the rarer genetic diseases of some people? How should our health dollars be spent? And if we make this permissible, on the one hand, I said it'd be too expensive, but on the other hand, we might advocate for it to be covered by public insurance for fairness, but then who makes the decision of when it's a high priority? Okay, so those were the limitations of public engagement. Now let's look at what the UK Nuffield Council produced, which were very important ethical principles. So we want to say too, we think those principles are necessary. And the principles that they emphasize, first of all, in Nuffield, they talked about respect for persons, entailing autonomy and respect for autonomy and protection of those with diminished autonomy. So that's a whole discussion in of itself, but basically how we need to respect people's choices over their own health. But now we're talking about germline gene editing for society. We also have to really emphasize what Nuffield said, which is social justice, inclusion, and solidarity. And so those are necessary principles. But here's the interesting thing, as someone who's taught bioethics and studied for a long time, and loves principles, because I'm a philosopher, it turns out principles really don't get us to good policy in and of themselves. They don't get us very far, in my opinion. First of all, internationally, the words and terms and even concepts are used to say different things in different countries and cultures. I've seen this a lot with attempts to govern AI, where privacy means something very different, for example, in China or the United States. So principles do not really give us the kind of contextualized, specific guidance we need. Second of all, even with any one culture, in any one group of principles, the principles themselves often compete. So for example, for rights of research subjects, we have a report, the Belmont Report, which protects human research subjects by emphasizing 3 principles: respect for autonomy, justice or appropriate inclusion, and beneficence, or making people's health better and not worse. And what we see is those 3 things can easily compete. We can have a medical treatment that would be ones that would respect people's autonomy and that might make them better, but that like this one, might cost too much for many people to have, so it doesn't meet the distributive justice criteria. We can have things that we can justly distribute, but that might not be sufficiently beneficial, or that might not sufficiently respect people's autonomy. For example, we could distribute some kind of gene edit that affects people with mental disabilities that might not be able to make the decisions for themselves. So we wouldn't be respecting autonomy in certain ways, even if we were doing something that we saw as beneficial for them or as something we could distribute equally. So basically, principles conflict and they don't give us clear guidance. And I just want to say quickly that in contrast, when we do contextualized case-based analysis, we can also do better in getting people from diverse religious and value and societal belief systems to agree, where they would never agree just on principles alone. And we saw that there was a US President's Commission on a number of bioethical issues in the 80s and late into the early 90s. And many times that was composed of people from diverse backgrounds, diverse faith traditions, diverse value traditions. And they basically didn't agree on anything until they would actually get into a contextualized case discussion. And then when they looked at the actual issues on the ground, they were able to make progress. And there's a lot written about that and how we needed context for people to move their thinking and come to agreement. Okay, so that's my critique with Lee and others in this article of ethical approaches, and leaves us wanting something more. But what more? Well, if we're going to say what more do we need besides principles and public engagement to come up with good policy for germline gene editing, GGE, let's again remember what are the specific ethical issues that we really need to talk about that are societal with germline gene editing. Well, first of all, there's the impact we've mentioned on people with disabilities and the further risk for stigma. So that includes people with cystic fibrosis, sickle cell disease, and deaf communities. And I'll just mention that this is fascinating, but in a lead article in one of our top journals, Sandra Sufian, who has cystic fibrosis, and Rosemary Garland Thompson, who has a limb development disorder, co-authored an article saying that they felt that their health differences were not making their lives lesser lives in any way, which of course they are not lesser persons in any way. And so they thought that to consider germline gene editing, both for a limb disorder and also for cystic fibrosis, was unethical on its face, because Sandy argued that her cystic fibrosis is like deafness. It's given her a different way to live her life, that we would lose that valuable difference if we did gene editing for cystic fibrosis. And I know Sandy, and I know she talks about she spends 3 hours every morning having to do very rigorous respiratory cleansing because of the deep mucus with cystic fibrosis. She has certain limited lifespan expectations, although much better than they used to be. And yet she has really found that her participation in the cystic fibrosis community and her awareness of vulnerability have really enriched her life and her work. So it becomes very, as you can see, these are very debatable topics. Another concern with germline gene editing is what we've talked about quite a bit, which is with the cost. They're very likely to increase health disparities. And in particular, we wrote about a concept that we came up with of intergenerational injustice, that we already know that people inherit wealth from their families, people inherit access to power within families, but now they would actually inherit access to basic health opportunities. And then the third area is the specter of eugenics, and that is a very big concern of many people. What if this was misused in terms of, for example, selecting people with higher intelligence, or even worse, for racist or other reasons? So what can we do? We have principles, we have public engagement, we need them both. Well, we suggested another important tool that we need is to consider this from a human rights perspective and do a human rights impact assessment. And I'm trying to look at the time, and we're not going to have that much time left, so I'm not going to spend a lot of time on this, but we definitely hope you'll read our article. And but a human rights impact assessment provides human rights are an international set of guidelines about how everyone in the world needs to be treated. And they're very important because they often are put into law. And they provide socially transparent criteria to examine political and socioeconomic power imbalances. So there are rights against being manipulated, rights against being controlled against our will, and rights to be included in the basic dignity and things we need to have a good quality of life. And they primarily try to protect the most vulnerable who may lack voice. So even though public engagement is a great thing, those people who might be too sick to engage, too poor to engage, and other people who lack voice may never even express their needs in the public, whereas human rights doesn't wait for people to participate to protect them. And when we think about a human rights impact assessment, it's not anti-technology or anti-progress at all. It looks to use any new innovative technology, including genome editing technology, to improve society's collective health while protecting core rights. So there's this long list of steps to go through, but I'm just going to go through 3 with you that apply to germline gene editing. So here's step 3. Determine whether the policy is well targeted. Is the intervention selectively inclusive in a way that doesn't exclude already marginalized individuals and groups who may benefit? And here are, again, our concerns about cost suggest that given the cost of germline gene editing in of itself, without a plan to change that, there will be people who need the gene edit just as much as people who get it, but who don't get it for economic reasons. So we're already seeing a problem there. Another criteria, sorry, here we go. Step 4, examine each policy for human rights burdens. And I won't go on and read the whole thing, but does it burden an already vulnerable group? And we just talked about how Sandy Sufian would argue that it would burden people with cystic fibrosis to have some people edited. The deaf community has argued strenuously that it would burden the deaf community. And the question is, what happens to people? Or what if we got one so far as enhancing IQ and people just with lower range IQs started to be devalued in society? And then a third, I'm sorry, must be going backwards again. Okay. And then determine whether the policy is the least restrictive alternative that can achieve the goal. And this is really important because one of the main, the former director of the NIH and I spoke also about this when it was happening. And Francis Collins, and one of the things he told me is that germline gene editing, the main group lobbying for it was people who were bilateral for the gene, couples who had bilateral, both of them had the gene for one gene for Huntington's disease, and saying that there's about 100 couples like that, he said, in the United States. And to change the whole world's policy on germline gene editing so that 100 couples could have a biologic child without risking Huntington's is a lot to do for a small group of families, I would argue, and you can push back in the Q&A, where there's also the option of those couples adopting children. And I understand that that's different. But the question is this the only way we can get there? Or we've thought about that with Tay-Sachs disease, where people could do screening in advance. So I think I'll stop there with this statement. A human rights impact assessment is needed to ensure that the same technology that could cure diseases like cystic fibrosis or sickle cell does not further marginalize people with disabilities. Sandy would argue cystic fibrosis should be included in that. Or does it create a form of social injustice in the form of aggregated inheritable health advantage for the most privileged members of society? And in some ways, that depends on how we govern it.